Right now, if you take a standard genetic screening test, the results you get back are, well, they're largely prioritized by a single, um, pretty major mathematical flaw.
Yeah, it's essentially just counting.
Exactly. I mean, for decades, medicine has prioritized genetic risks based on how common they are rather than how devastating they actually are.
Which is a huge distinction.
It really is. And I was looking at this graphic in the medical paper we're exploring today, and it just perfectly captures this flaw.
Oh, the scale visual.
Yeah, the scale. It shows the scale, but the fulcrum balancing it all is a twisting DNA double helix.
It's a great image.
It is. And on one side, you have this simple bar chart labeled population frequency, and it's basically just asking, how common is this genetic variant?
Right, just looking at the raw numbers.
But then pulling the scale in the completely opposite direction is a box labeled clinical impact.
And that have the icons, right?
Yeah, the icons of a heart, a brain, and a wheelchair. And that side asks a much, much heavier question. It asks what actually happens to a child if this gene is inherited.
Which changes the whole conversation.
Completely. So, welcome to the deep dive. Today, we are taking you on a mission to completely rethink inherited genetic risk, and we're basing this on a really groundbreaking paper called Beyond Carrier Frequency.
It is a profound shift in how we approach preventative medicine. I mean, really fundamentally.
It is.
The core equation that anchors this entire paper is elegantly simple, but it rewrites the rules.
Yeah.
It's just this: risk equals frequency multiplied by consequence.
Frequency multiplied by consequence.
Right. Historically, genetic screening has heavily favored that left side of your scale, the frequency side.
Just counting the variance.
Exactly.
Yeah.
But the authors of this paper, Dr. Boris Petrikovsky and Dr. Levny Motin, they argue that looking at frequency alone is, well, it's dangerously incomplete.
Yeah, I mean, obviously.
So to prove this, they use Jewish founder populations, specifically Ashkenazic, Sephardic, and Mizrahi groups, as this really well-documented model to build a brand new mathematical framework.
Which is fascinating.
It is. And it's a framework designed to eventually be applied to ancestry-linked groups all over the world.
Okay, let's unpack this. Because to really understand why adding consequence to the math is so revolutionary, we have to look at the history of how we've traditionally balanced these genetic risks.
They do.
And that history, it really begins with the Jewish community's response to some incredibly devastating genetic disorders, right?
It does. The historical roots of widespread genetic screening were driven by this urgent community-wide need to prevent severe autosomal recessive disorders.
And just to clarify for everyone listening, autosomal recessive simply means...
Oh, right. It means that a child has to inherit a non-working copy of a specific gene from both parents to actually develop the condition.
Right. So if you only have one copy, you are just a carrier.
Exactly. And the most famous catalyst for this widespread screening was Tay-Sachs disease.
Right.
It's a catastrophic infantile neurodegenerative disorder that was just absolutely devastating the Ashkenazic Jewish community.
It's heartbreaking.
It really is. Babies would appear completely healthy at birth, but then they would soon lose motor skills, experience seizures, and tragically pass away in early childhood.
The trauma of that on a community level, I mean, it must have been just unimaginable.
Unimaginable, yes.
And it led to two very different approaches to screening, as the paper outlines.
Right. The two models.
Yeah. So first, you had Dor Yeshurun, which operates on a confidential compatibility system.
Yes.
So prospective couples are tested, but they aren't, like, handed a printout of their genetic data, right?
No. They are simply told whether their match is genetically advisable, meaning they don't both carry the same recessive trait, or if it's a high-risk match.
Wow.
Yeah. And that model was incredibly successful because it was built around the cultural needs of the community, specifically avoiding the social stigma of being a known carrier.
That makes total sense.
Yeah.
But then on the other end of the spectrum, you see the development of open medical models.
Right, like J-screen.
Yeah, J-screen. And in that system, individuals receive their full, specific genetic results.
Yeah.
Exactly. Alongside genetic counseling to help them understand exactly what variants they possess and what that actually means for family planning.
Right. But the paper points out this massive blind spot that developed regardless of which model a community used.
It's a huge issue.
Yeah. Over time, the priority of what actually made it onto a screening panel was driven mostly by how common a carrier state was in the population.
Right. Frequency first.
Frequency first. But if we think about that practically, I mean, the logic just falls apart.
It completely breaks down.
Like, say you are running a mechanic shop.
Yeah.
You have a minor issue, like a scratched bumper. That happens to 1 in 10 cars.
Okay.
Then you have total brake failure, which only happens to 1 in 40 cars. You don't prioritize fixing the scratches just because they happen every day.
No, of course not. The focus has to be on the systemic threat.
Right.
And that exact problem is why the authors introduce what they call the Risk Priority Index, or the RPI.
It's the RPI. Okay.
Yeah.
Yes. The RPI provides a transparent, objective way to rank these conditions. You calculate it by taking the carrier frequency percentage and multiplying it by a severity of consequence score.
So you aren't just running a tally of how many people carry a variant.
No, you are weighing that frequency against the actual clinical gravity of the disease.
I have to pause you there because putting a number on human suffering, it just feels intensely subjective.
It is a really difficult concept.
Yeah, I mean, we know how to measure frequency, that's just counting.
Yeah.
But how do you objectively measure
It's
consequence?
a fair question.
Like, who gets to decide what a 4 is versus a 5? Isn't a score of 4 still completely life-altering for a family?
It is a vital question. But the authors, they aren't making moral judgments on the value of a life.
Okay.
They are creating clinical boundaries based on medical reality. The paper introduces this severity of consequence score, or SCS, which ranges from 1 to 5 based on objective medical criteria.
Okay, so what does a 1 look like?
A score of 1 means the condition has mild or low medical severity. So perhaps it requires occasional monitoring, but it doesn't impact lifespan or daily function.
Right, like a scratched bumper.
Exactly. Now, a 4 represents a severe disease requiring intense medical intervention.
Which is still a big deal.
It is. But a 5 is a distinct threshold of catastrophic outcome.
Okay.
We are talking about conditions that are uniformly fatal, profoundly disabling, or cause irreversible damage very, very early in life.
I noticed when reading it that they were very careful not to just label a level 5 as lethality.
Yes, and what's fascinating here is why they specifically reject lethality as the ultimate metric for consequence.
Why is that?
Lethality is simply too narrow of a lens. Consequence has to encompass conditions like deafblindness, severe bleeding disorders, or chronic organ failure.
Oh, wow.
Right. These are conditions that might not end a child's life immediately, but they impose a massive, lifelong medical burden and profoundly alter their physical reality.
So if a screening panel only prioritizes what kills, it just turns a blind eye to an enormous amount of preventable human suffering.
Precisely.
To put some real data behind this, the Ashkenazic risk priority table in the paper provides just a wild contrast.
It really highlights the math.
It does.
Yeah.
Let's look at Tay-Sachs disease again. The carrier frequency is relatively low, about 1 in 27 people.
Right.
But because it is a catastrophic infantile neurodegeneration, it gets a severity of consequence score of 5.0.
A maximum score.
Right. And when you multiply that out, it gives Tay-Sachs a risk priority index of 18.5.
Okay.
Now compare that to a condition called familial Mediterranean fever.
Yes, FMF. Familial Mediterranean fever has a massive carrier frequency in certain groups.
It's 1 in 5.
1 to 5.
1 in 5 people. If we were just using the old frequency-first spam filter model, familial Mediterranean fever would absolutely dominate the entire screening conversation.
Would push everything else off the table.
Exactly. But under this consequence-weighted framework, it gets a severity score of only 2.0.
Right. And that score of 2.0 is based on the underlying biology and the available treatments.
Because it's treatable.
Yes, FMF causes painful inflammatory episodes, but it is highly manageable with daily medication, like colchicine.
Okay.
Furthermore, it often has what we call reduced penetrance.
Meaning you can have the genetic typo, but your body somehow, works around it and you never actually get sick.
Precisely. Because of that reduced penetrance and the availability of really effective treatment, its actual clinical consequence is much lower.
So it gets a lower score.
Right. So, while its risk priority index is elevated purely due to the sheer volume of carriers, it doesn't artificially eclipse the catastrophic level 5 diseases.
This is such a crucial takeaway for you, the listener. This framework prevents common but treatable conditions from automatically drowning out the rarer, utterly devastating ones.
It keeps the scale balanced.
Yeah, it keeps our eyes on the brake failures, not just the scratched bumpers.
But, medicine is rarely that tidy.
Oh.
Yeah, as soon as you try to organize human biology into objective boxes numbered 1 through 5, you run into conditions that just refuse to cooperate.
That tension right there brings us to the absolute ethical curveball of this paper, which is hearing loss.
It is a massive challenge.
I had to read this statistic twice to make sure I wasn't, misunderstanding the data.
The carrier rates.
Yes.
Yeah.
1 in 6 people are carriers for mutations that can cause hearing loss.
1 in 6.
It is incredibly common.
But when the authors tried to assign a severity of consequence score to it, they hit a wall.
They did. And this raises an important question, perhaps the most ethically sensitive one in the entire source material.
It feels very charged.
It is. The challenge is that genetic hearing loss is a massive, imprecise umbrella term.
Okay.
On one end of the spectrum, you have isolated, non-syndromic hearing loss.
Meaning it's just hearing loss, nothing else.
Exactly. A person with this genetic variation might be completely healthy in every other biological respect.
With modern interventions, educational support, and a really strong deaf community identity, this is fully compatible with a rich, vibrant life.
But then on the other end of the spectrum, you have conditions like Usher syndrome.
Yes. Usher syndrome type 1F is a prime example here.
What does that look like?
It involves profound congenital deafness, but it is also accompanied by progressive vision loss leading to deaf blindness, alongside severe balance and vestibular dysfunction.
Oh, wow.
Alternatively, you might find hearing loss that is just one early symptom of a much broader, fatal neurologic disease.
So it's completely different.
You cannot assign a single mathematical score to all of those realities simultaneously.
And this is where I have to look at the ethics of the screening itself.
It's tricky.
Aren't we crossing a line here from preventing a fatal disease to just kind of selecting against human variation?
That is the core debate.
Right, like if a couple finds out they might have a child who is deaf but otherwise perfectly healthy, and they use screening to avoid that pregnancy, aren't we making a societal value judgment on the deaf community?
The authors are acutely aware of that tension. It's a huge consideration.
How do they handle it?
Their resolution is that these broad categories must be fundamentally dismantled in the medical literature.
Dismantled?
Yes. You cannot treat a 1 in 6 carrier frequency for a broad hearing loss panel the exact same way you treat a 1 in 6 frequency for a uniformly fatal infantile leukodystrophy.
Right, because the consequences are so vastly different.
Exactly. To respect both the prevention of severe disease and human dignity, the paper argues you have to clinically subdivide these categories.
Break them apart.
You must separate non-syndromic hearing loss from progressive deaf-blindness syndromes on the screening panels.
That makes a lot of sense.
If you just label it all hearing loss, you risk eugenic implications on one side and failing to prevent immense medical suffering on the other.
That same danger of applying a broad, clumsy label to a complex condition, it also applies to how we label the people actually taking these tests.
Yes, population labels.
Yeah, this paper argues passionately that populations shouldn't be lumped together either.
This requires us to look really closely at terminology, specifically the word race.
Okay.
The paper operates with incredibly strict parameters here. In this scientific context, race is used exclusively as a descriptor of historically formed ancestry groups.
It's just about history.
It acknowledges historical realities like endogamy, which is the practice of marrying strictly within a specific community, as well as founder effects and genetic drift over centuries.
So it's never used as a social hierarchy.
Absolutely not. It is never a cultural identity or a claim of biological purity.
Okay, because I always just assumed Sephardic was a unified category for screening purposes, like you check a box.
A lot of people do.
But here's where it gets really interesting. When you look at the Sephardic and Mizrahi risk priority table in the text, you realize that it does not describe a single genetic risk pool.
Not at all. It is a dangerous oversimplification.
How so?
We have to look at the historical mechanisms of population bottlenecks. Imagine a small group of founders migrating to a specific region, say Libya or the Caucasus Mountains. For centuries, due to cultural and geographic isolation, they practice endogamy. Whatever rare genetic quirks those original 50 or 100 founders had, they don't get diluted by a wider population.
Because they're only marrying within that small group.
Right. So those genetic variants get amplified over generations. That is a founder effect.
And because those migrations went to different places, the genetic drift was different for each specific group.
Precisely.
Let me give you some examples from the paper because the severity here is just intense.
That's eye-opening data.
There is a condition called aryl sulfatase, a deficiency, which is a severe leukodystrophy.
Meaning it progressively destroys the white matter in a child's brain.
Yeah. And in Habonite Jews, the carrier frequency is 1 in 6.
which is extremely high.
And it has a massive chart-topping risk priority index of 83.3.
It's huge.
But if you look at Libyan Jews, that's not their primary risk at all.
No, it's completely different.
Their highest signal is for limb girdle muscular dystrophy with a carrier frequency of 1 in 10. And then you look at Dagestani or Caucasian Jews, and they have a high signal for MED17 progressive microcephaly.
Where the brain simply stops growing properly.
Exactly, and that has a frequency of 1 in 20, so they are all different.
Yes. So if a modern screening program just tested a Moroccan Jewish individual for the Habonite Jewish variants, simply because they are both categorized under a broad Sephardic label, the test would be functionally useless.
Wow.
The genetic architecture is completely different based on their specific geographic isolation.
It's like trying to navigate your GPS by broad time zones instead of exact zip codes.
That's a great way to put it.
Right. If you just say, hey, I'm in the Eastern time zone, and you rely on a map built for that broad label, you are going to drive right off a cliff because you missed the local terrain.
You really are.
You have to narrow it down to genetic ancestry, family origin, and specific founder lines to get a screening panel that actually protects people.
Broad group-level language is a starting point for a conversation, yes, but it must be refined into actionable, granular descriptors before any clinical decisions are made.
All right, so we've established that we need the right math, right? The consequence-weighted formula.
Yes.
And we need the right population data down to the exact genetic zip code.
Correct.
But what exactly happens at the biological level when these genes actually meet? Because the paper shatters another major assumption I had.
The identical match assumption.
Yes. I always thought autosomal recessive risk meant that both parents had to have the exact identical mutation in their DNA.
It's a very, very common misconception.
Yeah.
We often say a couple is at risk if they carry the same recessive disease.
Right.
And while that's practically useful for a baseline explanation, the biological reality is much more complex, and it centers on a concept called pathogenic biallelic convergence.
Okay, let me try to puzzle that phrase out. Pathogenic means disease-causing.
Correct.
Biallelic means 2 alleles, so 1 copy of the gene from mom, 1 copy from dad. Convergence. Are they saying the parents' genes converge to cause the disease in a way we didn't expect?
You've hit the nail on the head. The critical event isn't necessarily that you inherit the identical spelling error in the DNA from mom and the identical spelling error from dad.
Okay, so what is it?
What matters is that both parents pass down a mutation that breaks the functioning of the same overall disease gene.
Even if the errors are different?
Yes, and this is formally called compound heterozygosity.
Okay, so it's like a car engine.
Let's hear it.
It's like if mom breaks the car's engine by removing the spark plugs and dad breaks the car's engine by pouring sugar in the gas tank.
I like this.
They didn't make the exact same error, but the result converges on the exact same outcome.
The engine won't start. The child inherits no functional copy of that gene.
That captures the mechanism perfectly. And this distinction is vital for how we design screening systems.
How so? Well, if a compatibility system only looks for an exact marker-to-marker match, meaning they only scan to see if mom and dad both remove the spark plugs, they're going to miss the risk completely if dad actually poured sugar in the gas tank.
Oh, wow.
Both variants converge on the same functional unit, causing the disease, even though the parental markers weren't identical.
That is deeply unsettling if you're relying on a basic older generation screening panel.
It is. And the biology pushes even further into a realm called digenic and oligogenic inheritance.
More big words. What does that mean?
This is where you have variants in entirely different genes that interact to disturb a biological pathway.
Wait, so instead of breaking the same gene, they are breaking different parts of a larger system.
Yes.
It's like a bridge.
Okay, go on.
Mom passes down a genetic variant that weakens the suspension cables. Dad passes down a variant that weakens the concrete pillars.
I see where you're going.
Separately, the bridge still holds the weight of traffic, but together, the whole thing collapses into the river.
Yes, exactly. The variants interact across different genes to cross a physiologic threshold, causing the collapse.
That's wild.
It is, however, the authors are very careful to caution that these cross-gene interactions are much less predictable than classical autosomal recessive inheritance.
Meaning we can't always know if the bridge will collapse.
Right. They require a much higher standard of clinical evidence before we should start using them in reproductive counseling. We can't just treat them like a standard 1 in 4 recessive risk without rock-solid data proving that the bridge will actually fall.
So what does this all mean for you listening right now?
It's a big takeaway.
It means that if we rely on screening systems that only look for exact, identical marker-to-marker matches, we might be handing couples a false sense of security. The science is telling us we need to look at the whole engine, not just the spark plugs.
It demands a screening architecture that is as nuanced as the cellular biology it's trying to interpret.
Let's recap the journey we've taken today because we have covered incredible ground.
We really have.
We started by moving away from the old, flawed model of just counting carrier frequencies, learning instead how to calculate actual consequence using the risk priority index.
The RPI.
Right? We waded into the deep ethical waters of variable conditions like hearing loss and why we have to respect both disease prevention and human identity by dismantling broad categories.
Crucial step.
We broke down the myth of the monolithic ethnic group, exploring how historical bottlenecks mean a broad label like Sephardic just isn't enough to protect specific founder lines.
The zip codes, not time zones.
Exactly. And finally, we looked under the hood at the deep biology of compound heterozygosity, realizing that two different genetic errors can still cause the exact same tragic result.
If we connect this to the bigger picture, it is vital to remember that this paper is just part one of a much larger mission.
Part one.
Yes, the authors used the Jewish founder population data because it has this meticulous history of genealogical tracking and distinct geographic isolation.
It's the perfect model.
It is. But part two of their project will take this consequence-weighted framework and apply it to other ancestry-linked groups globally.
Oh, wow.
Every population has its own unique pattern of founder variants, carrier frequencies, and community-specific ethical concerns. This mathematical and biological framework is designed to be portable, helping communities worldwide make clearer, more compassionate screening decisions based on impact, not just volume.
Which leaves me with one final provocative thought for you to ponder.
Let's hear it.
We've talked so much about the severity of consequence score, ranking diseases objectively from a mild 1 to a catastrophic 5.
Right.
But medicine is not static. As CRISPR, gene therapy, and other radical medical technologies advance, once fatal diseases are becoming treatable.
They are.
So how will our consequence scores shift over time? Will a disease like Tay-Sachs, which scores a devastating 5 today, become a manageable 2 in 50 years?
It's entirely possible.
And if it does, how will that constantly moving target reshape the way humanity handles genetic screening for our future generations?
The math will have to change.
The weights on the scale are always changing. Thank you for joining us on this deep dive.